I am going to first share the PowerPoint and then I'll get started. And I'm starting with the first study outline from the, like if you go in the content area, the first Unit 5 section, which, let me see, it's just called the urinary system. So I will, here we go. I'll be starting with that study outline, and then there's a second one in the other content area for the urinary system for within Unit 5, which covers more on like urine formation and then body fluids. Okay. So I am sharing the PowerPoint for the urinary system. And I am just going to go through like I've been doing, going through the lecture outlines. But there are, I don't know if you guys have looked, but there are several pre-recorded lectures that I have already posted in especially in that first Unit 5 content area. There's 3 of them, which are really, really helpful for some of the physiology. When we get to those sections, I'm not going to do a whole other lecture on them for the sake of time, because I want to get through as much stuff that's not covered by those pre-recorded lectures. I want to get through all of that with you guys today, but I'll let you know when we get to that section that there is already a lecture that's been recorded for that, and I'll skip over that section and then just continue with the rest of the outline. Okay. So the very first thing on the outline is just a review of the urinary system itself, what structures are involved, what the main functions are. So basically the kidneys are the main organs that are doing the work of the urinary system. And really what they're doing is filtering your blood to remove any excess substances, any waste products, any harmful byproducts of metabolism, for instance. So the organs and structures that are involved with the urinary system are listed. First thing in your outline, there are the 2 kidneys, which are the actual organs that filter blood to produce urine. And you can see those organs here on the PowerPoint. They are the brownish structures. Then you guys can see, hopefully, the 2 tubes that extend down from the medial side of each kidney. These are the ureters. These are paired tubes that carry urine from the kidneys to the bladder. So if you look at the diagram, you can see that each one extends down from the medial side of the kidney right here, and it extends all the way down to the bladder, which you can see down here. So that is the next structure. The urinary bladder is simply a storage area for urine. And then finally, the urethra, which you can't really see too well down here, but it's a shorter tube, a single tube that carries urine from the bladder to the outside of the body. Okay, and then major functions of the kidneys. They are just listed here. They're listed in your outline. So the first thing you guys probably think of is like removal of waste from the body, but that's actually a secondary function. Really, the main job of the kidneys is to regulate what's in your blood. And they help to regulate blood volume. And because blood volume is directly related to blood pressure, it also regulates blood pressure. This is a big thing that regulates blood pressure. So basically by removing fluid from the blood, it's helping to regulate blood volume and blood pressure. Also, it helps to regulate the concentrations of various substances within the blood, different ions like sodium, potassium, also nutrients, like glucose, amino acids, etc. It also helps to regulate the pH of blood. Remember that the main ions that influence blood's pH, particularly if you have an accumulation of them, they make the pH go down. Those are hydrogen ions. So, if blood's pH is getting too low, then the urinary system can just remove more hydrogen ions from the blood and send them out of the body in urine. Also, you may not have known this function, but the kidneys do produce hormones and enzymes. So that's another function. And then just a byproduct from the first three functions is the excretion of wastes. So in doing those first three functions, regulating blood volume, regulating various substances in the blood, regulating blood's pH, it's going to then remove anything that was in excess. And it's going to excrete it from the body as urine. Okay. So as far as kidney anatomy, the kidneys are retroperitoneal, which means that they're actually located behind and outside of the peritoneum. If you remember when we talked about the digestive system, that is the huge, it's the biggest membrane in the body, the huge membrane that surrounds just about every organ that's in the abdominopelvic cavity. But the kidneys are not within that membrane. They're located behind that membrane. So they're retroperitoneal. Just looking at the kidneys, I'm gonna go, to this diagram. You can see some of the things that I'll be talking about here. External anatomy, the hylus is the depression here on the medial side of each kidney. Actually, I'm gonna go back here, or you can see the kidneys just from the outside. So it's the medial depression on this side, the medial side of the kidney, where the ureter and where blood vessels enter and exit. Same thing for this kidney. And then we talked about this when we had lab on Tuesday with the first group. I'll have lab again this coming Tuesday with the other group. So we're doing the kidney dissection. So we'll look at some of the things you can see on the outside, and then we cut into the kidney. One of the things that I explained is that there are 3 layers of tissue that normally surround the kidney. The outer 2 layers will have been removed from the kidneys that you guys are dissecting, but you will get to see the innermost layer. There's not a whole lot to see, but it is on there. So these layers are listed from outer to inner. The renal fascia is the outer layer. And it's just really dense connective tissue that holds the kidneys in their place. They attach the kidneys to the back body wall. So it helps to keep the kidneys attached or anchored in their location. Then just inside of that layer is the adipose capsule. Remember that adipose tissue is the tissue that stores fat. So this is the middle layer that is a fatty, it's a pretty thick fatty layer. And that helps protect the kidneys from trauma, from physical trauma. Typically,
can you repeat that one more time? I'm sorry.
Yeah, just it's the fatty layer that protects the kidneys from physical trauma. And I was just going to say that typically with any important internal organ that you want to protect, you are going to have a layer of adipose tissue around it for like cushioning and protection. Okay, and then Finally, the innermost layer is called the renal capsule. This is the only layer that will still be attached to the kidneys that you guys are dissecting. It's the innermost layer, and it's a very thin, transparent membrane that contains white blood cells to help prevent infection. Okay. So as far as the blood supply to the kidney, you definitely should know the renal artery is the artery that carries blood, oxygenated blood into the kidney. You can see that here on this picture, that big red artery going into the hylus, and that the renal vein is the blood vessel that carries blood out of the kidney. And this is now deoxygenated blood. You do not need to know these intermediate arteries that are listed here, segmental artery, interlobar artery. You don't need to worry about that. But then we are, in probably just a few minutes, we are going to talk about the two arterioles within the kidney and then the two capillary beds. So you should be familiar with those, and we are going to talk about those in just a little bit. Okay, the rest of the things. So basically, like if you're looking at the PowerPoint, what's in the green regions, like here and then down here, these are the blood vessels that you need to know. The other ones, no, you do not need to know. So when we cut into the kidney, which this is exactly like the same type of cut that you're going to make when you do the dissection, or when, if you already did it, when you did the dissection, it's going to look something like this. And this allows you to see into the kidney. So you can see the various regions and the different structures that are in here. So first of all, there are two distinct regions within the kidney. The renal cortex is this outer portion. And you can see on the diagram, it does look lighter. And even on the actual cadaver photo, it looks like a lighter color. That's how you can identify the cortex. So it's the outer region. And then renal columns are part of the cortex. The renal columns are extensions of the cortex. That project or extend between each pyramid. We haven't talked about the pyramids yet, but we are about to. If you're looking at the diagram, the renal columns are identified, and I'll point them out. They are these right here, these extensions of the cortex that go in between these triangular structures, which are called the renal pyramids. So here and here, here and here, those are the renal columns. Okay, the other main region of the kidney is called the renal medulla. That's the inner region. And there are several structures that are located here. They are all listed in your outline. You have, first of all, the renal pyramids, which are these right here, these triangular structures. They look like pyramids, so that's how they got their name. Okay, these darker triangular structures. The renal papilla is the opening at the end of each pyramid. So it's at the very tip of each pyramid. That's where the renal papilla is located. Okay, and then there are two sets of channels that lead off of each pyramid. The first one is called a minor calyx. The second is called a major calyx. So A minor calyx is a channel leading off of each pyramid. Okay, so these little channels here, right here, right here, right here, that come off or just extend down or off of each pyramid. And then a major calyx is formed, I'm sorry, I should say, first of all, it's a larger channel that's formed when several minor calyces unite. And I'll identify that on the diagram in a sec. So it's a larger channel that's formed when several minor calyces come together or unite. So if you guys can look at the diagram, there are minor calyces coming off of pyramids here, here, and here, and then where they all kind of empty into, that forms a major calyx. So minor calyces drain into major calyces. And then finally, the renal pelvis is the larger open region in the middle of the kidney. And it extends directly into the ureter. So when urine is formed within the kidneys, it first comes out of the renal papilla, which are the openings at the tip of each pyramid. And from there, urine is going to drain into a minor calyx, then into a major calyx, and then finally into the renal pelvis, which is right here. And then from there, in order to leave the kidney, it goes through the ureter. And that ureter, remember, is the tube that extends out of the kidney and carries urine down to the bladder. One last thing here, the renal sinus is this entire area. It's a cavity that contains the renal pelvis and all of the calyces. So the renal sinus is this large area, which is a cavity. If you were to dig out all of these channels, all the calyces and the renal pelvis, you would see just kind of a cavity here in the middle. So it's the cavity that contains all of the calyces plus the renal pelvis. Okay, so that is all that you can really see when you're looking at the outside and the inside of the kidney. But then when you're inside the kidney, there are actually more structures there, which you can't see with the naked eye. These are structures that are microscopic. So you will not be able to see these when you do the dissection, but they are super important. These are really the things that are performing the main function for the kidneys. These are called nephrons. That's what you're looking at right here. This is a nephron. There are millions of these within each of your kidneys. And basically the nephron is the functional unit of the kidney. This is what's actually allowing the kidneys to perform their function. For the location, these are found primarily in the renal cortex and in the renal pyramids. Another interesting thing is that you are born with a certain number of nephrons, and unfortunately, if they are damaged, they cannot be repaired, they cannot regenerate. So if you have major kidney damage, it's going to stay like that. It can't be repaired. Also, if you look at the nephron, there are two distinct areas. This first area right here is called the renal corpuscle. The rest of it, this long twisted tube, is called the renal tubule. And then the function of the nephron, which you can fill in on your outline, there's a space for that, is that they filter blood. They filter blood to produce a fluid called filtrate. And that gets processed by the nephron to form urine. Okay, so now we're going to take a closer look at the nephron and look at the individual parts within the nephron. I did say, remember that there's two main regions, but within those regions, there are particular structures that we'll learn the names of. So we have, remember, first of all, the renal corpuscle is this first section here. The rest of the nephron is called the renal tubule. So we'll look at the renal corpuscle first. This is the site of filtration. This is where blood gets filtered. And it's done here. It's by these, done primarily by the glomerulus, and then the filtrate is going to go into the glomerular capsule. And those are the two parts of the renal corpuscle. So first of all, the glomerulus is the red, you guys see the red blood vessels in here? That's it. The glomerulus is a mass of capillaries that's specialized for filtration. Okay. You have a couple more details in your outline, like these capillaries have little holes in them. They're fenestrated, which means they have holes in them, which make them very porous, which is good because that allows substances to leave the capillary and enter the renal or the glomerular capsule. This is the only capillary bed in the body that is supplied and drained by an arteriole. Usually you have an arteriole carrying blood into a capillary bed, and then you have a venule draining blood out of a capillary bed. But in this case, you have an arteriole that's actually draining blood out. So that's a little bit unique. The other component of the renal corpuscle is the glomerular capsule. So if you kind of take the renal corpuscle and you isolate it, this is what it looks like. Here's the glomerulus, that's the capillaries in the middle. And the glomerular capsule surrounds it. So what you can add in here for just a description of it is that it's a cup-shaped structure that surrounds the glomerulus. And there is a parietal layer and a visceral layer. The parietal layer is the outer wall, which you can see out here and here. So it's the outer wall of the capsule. The visceral layer is the inner layer that directly touches the glomerulus. So from this picture, you can't really even tell that it's there, but it directly covers these capillaries. If you kind of take an enlarged view, here is the capillary. Okay, this is the capillary. You can see all the little fenestrations, all the little holes in it to allow substances to leave the blood. And then this here surrounding the capillary is that visceral layer of the glomerular capsule. It just wraps around the capillary. So this layer, that visceral layer, is composed of cells that are called podocytes. And those cells have what are called pedicels, which are thin extensions. That's actually what you're seeing here. These are thin extensions that wrap around the capillaries of the glomerulus. So these extensions wrap, kind of like fingers, they wrap around the capillaries. And looking at this diagram, you can definitely see that. You can see these finger-like extensions that are wrapping around the capillary. In between those extensions are little tiny spaces. These are called filtration slits. These are the spaces between the pedicels. So those openings, those slits, also allow substances. Once they've left the capillary, they allow substances to get through into the capsule. And looking at this picture again, where you can see the glomerulus and the capsule surrounding it, in between those two layers of the capsule is a space that's called the capsular space, in between the parietal layer and the visceral layer. And this space fills up with filtrate during the filtration process. So it's into that space that filtrate is going to go as soon as it's produced. Okay, so now we're done with that first section, the corpuscle, and now we're going to move on to the renal tubule. This is the site of the two other processes that are involved in urine formation. So remember, filtration occurs at the renal corpuscle. The other two processes are called reabsorption and secretion. So this is where those two processes occur. This first part here that's circled in green is called the proximal convoluted tubule. And it's kind of a long name, so we often just call it the PCT for an abbreviation. It's the very first part of the tubule. And for the histology, remember histology is like the tissue makeup of an area. So this is composed of simple cuboidal epithelium, so little cube-shaped cells. And they have on their apical surface, their exposed surface, they have microvilli. And that term should sound familiar. We learned about villi and microvilli when we did the digestive system. Whenever you have villi and or microvilli, they are involved in absorption. And this is where almost all reabsorption, which is substances actually leaving the filtrate and going back into the blood, most reabsorption happens here. So that's why you have microvilli here. They are good for that. They help with that. The next part of the tubule, this long piece that kind of hangs down, that's called the loop of Henle. There is a descending limb where fluid is flowing downward, and then an ascending limb where it's flowing back up. And this is the section of the nephron that helps determine how concentrated urine is going to be. We'll talk about that process a little later. And then the next two parts of the tubule, we have the distal convoluted tubule. We often call that the DCT. For the histology, it's the same as the PCT, except without the microvilli. So it is just simple cuboidal epithelium. But there's two modifications here, two special types of cells that are found here. These cells are called principal cells and intercalated cells. The principal cells help maintain the body's water and sodium balance. And the intercalated cells help to maintain the acid-base balance of blood.
Can you repeat that one more time?
Yep, they help maintain the acid-base balance of blood. Okay, and then the last section of the nephron is called the collecting duct. This has the exact same histology and special types of cells as the DCT. So everything's the same, simple cuboidal epithelium. It also contains those two special types of cells, principal cells and intercalated cells. And this part, this collecting duct, extends all the way down to the tip of the renal pyramids. Remember, that's where the opening is, called the renal papilla. So filtrate, as soon as it's formed, it's going to be passed all the way through the renal tubule, through all those parts that we just talked about. And by the time it gets to the end of the collecting duct, it's going to have traveled all the way down to the tip of each pyramid. Once it leaves the collecting duct, it's now referred to as urine. As long as it's in the renal tubule, it's called filtrate. But once it leaves the renal tubule, once it comes out of the end of the collecting duct, now it's called urine. And then it's going to drain out of that little opening, the renal papilla, and go into the calyces and into the renal pelvis. And then from there, it goes into the ureter and then into the bladder for storage until it's excreted. So now you have the whole process of how urine is formed and where all the structures that it's traveling through before it gets to the outside of the body. All right, now that we've talked about nephrons, you guys have information in your outline already filled out about the two main types of nephrons. There are cortical nephrons, which are the most common type. They're called cortical nephrons because the majority of the nephron is in the renal cortex, that outer region of the kidney. Just a little bit of it, like the bottom of the loop of Henle, dips down into the pyramids, but most of it's in the cortex. But the other type of nephron are called juxtamedullary nephrons. It's a long name. And these are the ones that are a little bit lower down. So they extend more deeply into the renal medulla. They're going down into the renal pyramids. You can see the loop of Henle is a lot longer and it's going much further down into the pyramid. So those are the two types of nephrons. Now we're going to look at the two capillary beds that are found associated with each nephron. The first one we've already talked about. The first one is the glomerulus. So that's here. Remember, the glomerulus is just that mass of capillaries that's part of the renal corpuscle. That's where filtration occurs. Okay, so that one we already discussed. That's here. Here's the glomerulus. Here's the glomerulus. The other capillary bed, it's called the peritubular capillaries. So these capillaries branch off of the efferent arteriole. And I will type that for you. They branch off of the efferent arteriole. If you're looking at the diagram, here's the efferent arteriole. It drains blood out of the glomerulus. So then you have this other capillary bed that then comes off of that arteriole. And if you look at this picture, that capillary bed kind of wraps around the outside of the renal tubule, especially around that loop of Henle. It kind of covers, it wraps around the outside of the renal tubule. And just like the glomerulus, these capillaries are very porous. That means that they just have a lot of little holes in their walls, which allows them to absorb substances from the tubule. So if there are substances that initially enter the nephron and now they're in the filtrate that's passing through the renal tubule, and these are substances that your body actually wants to keep, it doesn't want them to be excreted, then they get reabsorbed. So those substances can leave the tubule and move right back into the bloodstream by going into these capillaries that are directly covering the renal tubule. That's why we have them there, so that substances can move out of the tubule from the filtrate and go into these capillaries. In the juxtamedullary nephrons, these are the ones that are really long. So here's one over here. The peritubular capillaries have a special name. They're called the vasa recta. So that's written here, and that's written in your outline as well. Okay. I'm going to give you two more definitions, which come on, go to the next slide, hold on, come on the next page. There we go. And then oh no, I'm wrong. I'm not quite to the section yet where I have a recorded lecture. I thought I was getting there, but I'm not just yet. So never mind, keep going. There's a structure called the Juxtaglomerular complex. And it's located where a certain part of the tubule, the distal convoluted tubule, actually touches the afferent arteriole. So the arteriole and the tubule just come in contact right in this one spot. And it's right there that there are two special types of cells. There's a special type of cell in the tubule right at that spot. And there's a special type of cell in the arteriole right at that spot. These special cells are listed in your outline. These are the macula densa and the granular cells. So first, the macula densa cells are tightly packed epithelial cells that monitor the ion concentration of the filtrate. Okay, so these cells are keeping a close eye on the amount of ions that are in the filtrate, like sodium, potassium, chloride, calcium. Okay, and then the other cells, the granular cells, these are smooth muscle cells that monitor blood pressure in the afferent arteriole. So these monitor blood pressure in the afferent arteriole. Also, they secrete an enzyme that's called renin. I will type that in here for you. Renin, R-E-N-I-N. Okay. So the next section is to take a closer look at the 3 processes that are involved in urine formation. So we have, first of all, glomerular filtration, which is when the kidneys filter the blood to produce filtrate. Next is tubular reabsorption, which is when water and solutes are just any solid particle. It could be nutrients, it could be ions. They are reabsorbed from the filtrate back into the blood. So these substances move from the filtrate back into the blood. And then finally, tubular secretion is the movement of wastes or excess ions from the blood into the filtrate. Okay, and then after that last process, everything that remains in the filtrate is going to ultimately become urine and be excreted in urine. Okay, so we're going to talk about filtration in more detail. So filtration is, just first to give you a definition of just in general, what is filtration? It is a passive process. Basically, any process in the body could be passive or active. If it is an active process, it requires energy to happen. If it's a passive process, it doesn't. This is a passive process where pressure forces fluids and solutes across a membrane. Okay, so the membrane that we're talking about here is called the filtration membrane. And the filtration membrane is basically the wall of the glomerulus, which is the capillary, and the glomerular capsule, the inner layer of the glomerular capsule. So there are, and if you guys look at this picture, here's the capillary. These 3 layers form the filtration membrane. So blood is flowing through the inside of this, and it passes through these 3 layers, 1, 2, 3. So the 3 layers are listed, and I'll just describe them in your outline. First of all, the endothelium of the glomerulus. This is just the wall, the capillary wall. And it contains pores. If you guys look, it contains all these little holes. They allow everything in blood except blood cells to pass through. So everything in your blood except for your blood cells can cross that first layer of the membrane. Then the second layer is this layer shown in purple, that's the basement membrane, which surrounds any capillary bed. So the basement membrane has actually a negative charge, which allows everything except large, negatively charged proteins to cross. So everything except large, negatively charged proteins can get across this second layer. And then the last outer layer is the inner layer of the glomerular capsule, the visceral glomerular capsule. And remember that this layer contains filtration slits, which you can see here. It contains filtration slits that allow water and small solutes across. So any large molecules cannot fit through. Okay, so what gets filtered out of the blood and moves across this filtration membrane is called filtrate. So if you go down to below the picture in your outline, you'll see the definition of filtrate. Filtrate is the fluid that was produced from filtration, and it contains everything that was in blood except for blood cells and proteins, because either they're too large, or in the case of the proteins, they're negatively charged, so they were held back as well. Everything else that was in the blood gets across that filtration membrane initially. So you can see all of the things that filtrate contains. It contains water, it contains nutrients, it contains lots of ions. Okay, so let's talk about the forces that are causing filtration to happen. Net filtration pressure is basically the pressure that's responsible for filtrate formation. And it involves two main forces: any outward pressure, which is pushing across the filtration membrane, so it's promoting the formation of filtrate. And then there's inward pressures, which actually push in the opposite direction and would prevent the formation of filtrate. So those outward pressures are just one, hydrostatic pressure within the glomerulus. And basically that's just a fancy way of saying blood pressure within the glomerulus. This is just the actual blood pressure in the glomerulus. So that's the major force that is pushing blood out of the glomerulus and causing the formation of filtrate. That pressure is usually about 55 millimeters of mercury. Millimeters of mercury is that mmHg that you see, and that is those are the units that are used for pressure. There are two inward pressures, which are actually pushing in the opposite direction, which would kind of prevent the formation of filtrate. These are hydrostatic pressure in the capsular space. So this is just the pressure exerted by filtrate that's already been produced. So this is the pressure exerted by filtrate that's already been made, and that is in this capsular space here. So it's pushing in the opposite direction. But fortunately, it's not pushing with a whole lot of force, just about 15 millimeters of mercury. So not a big force at all. And then the other is colloid osmotic pressure. This is the pressure exerted by proteins in the blood. So they also kind of push in the opposite direction with a little bit more force, about 30 millimeters of mercury. So if we want to figure out the net filtration pressure, we take the pressure that is promoting filtration. So that's the blood pressure in the glomerulus, which is about 55. And then we have to subtract the two opposing pressures, which in total are about 45. So 55 minus 45 is 10. So you have an overall like net filtration pressure of about 10 millimeters of mercury. And your body is always working to maintain that constant, steady filtration pressure so that your body is constantly filtering the blood to produce filtrate. You never want that pressure to get too low because that means that blood is not being filtered. And that could mean that wastes are potentially building up in your blood, which could cause all kinds of problems. Okay, so the glomerular filtration rate is the volume of filtrate formed each minute by both of your kidneys. And like I said, this is always, your body's always working to maintain a pretty constant glomerular filtration rate. You don't want it getting too low because then you're not properly filtering your blood. You also don't want it getting too high because then you could be filtering too much. So there's like a perfect balance here that your body tries to maintain. So actually, this is a great spot to take a break because now we are at the point where I have a recorded lecture for this whole next section. So let's take a quick break. It's 10:23. So let's meet back at 10:30 and we'll pick up after the section that I have the recorded lecture for. Okay, so you have about 7 minutes. Take that break, do whatever you need to do, and I will meet you guys back here at 10:30.